Hello and welcome to today's tutorial where we're going to be exploring biology. Now biology is a study of living things and we're going to be exploring today one chemical that exists in every single living thing, and that's called DNA. DNA is a tiny chemical that's inside every single part of our body. And it's the recipe book to explain how to build a living thing. To help my DNA has all of the instructions on how to build me. If I cut myself, the DNA has all the instructions on how to build new skin cells. And the same with any other part of growing or repairing your body. Your DNA is really important. It's not just people that have DNA. We have DNA in all plants. A beetroot plant has the DNA to show the plant how to build beetroot. A spider has spider DNA. A caterpillar has caterpillar or butterfly DNA. And it's in every single living thing in the whole world. Today I'm going to show you how you can explore DNA and have a look at what it's like. So the equipment you're going to need is a tub or a bowl that can have hot water in it, and we're going to make a water bath using this. You're going to need a couple of glasses. In one of the glasses you need 100 millilitres of water. In another glass you're going to need about 100 millilitres of icy cold strong white rum or icy cold methylated spirit. Because we're using those chemicals, you're going to need a grown-up to help you with this experiment. We're going to be using hot water and we're going to be using strong alcohol. So you'll need somebody to help you with health and safety. So we've talked about water, rum. You're going to need a measuring spoon or a teaspoon, a fork and a spoon, some shampoo or shower gel or soap, any type of detergent. But the important thing is it needs to be completely colourless so that the colour doesn't block your view of the DNA that you're going to extract. Some salt, not a huge bag like this, just need a tiny bit, half a teaspoon of salt, and the kiwi fruit. And this is what we're going to see real life DNA of a kiwi fruit. It shows the plant how to build a kiwi, how to build a leaf, how to build a stem, how to build a root, and how to build a fruit. So first you're going to need to take off the skin from the kiwi because it's really hard and we're not going to get much DNA out of that. So you need to take the skin off and chop it up like I have here. And then you need to mash it. Now the more you mash it, the better this experiment will work. If you've got a pestle and mortar, you could use that to really grind it up. Or if you've got a handheld food processor, you could get your grown-up to whiz it a little bit in that food processor to really break it up. So mash away. You'll need to do it for longer than I have. I'm just going to do it quickly to show you what to do. So you've got your really mashed up kiwi here. So you've already broken it into tiny chunks, which has made the surface area much bigger. And that's really important because it's going to help the DNA to be extracted. So now you take your glass of water and you're going to add to that water half a teaspoon of salt, and that's all you need. Add it and then stir it until it's dissolved. You know that it's dissolved because you can't see it anymore. So keep stirring until you can't see any salt anymore. It doesn't mean that it's disappeared. The salt is still there, but it's just dissolved into the water. We really need the salt because it's going to help to extract the DNA from the kiwi fruit cells that it's in. There we go, that's dissolved now, I can't see it anymore. And then you need to put that on the floor so that it doesn't fall over. And then we're going to need 1 teaspoon of your detergent. Now a detergent is anything that's soapy. So I've got shampoo, you can use soap or shower gel, and remember, needs to be clear with no colouring. That'll make it work a lot better. If you haven't got any soap or shower gel that's clear, you could have a go with what you've got, but it's much, much better if it is see-through and you get the best view possible. Now, you're also going to dissolve this detergent in the salty water, but don't stir it too much because we don't want to make lots and lots of bubbles. So just stir it gently until it's all mixed in. Okay, now we've got an extraction buffer. That's a very scientific word, isn't it? It basically means a mixture of salt and water and soap. That's your extraction buffer. And you're going to add your mashed up kiwi fruit to that. Now what this is going to do is it's going to break open the kiwi fruit cells. And the DNA that's trapped inside will go out and become floating around in that mixture. Now, this is a very important part now. You're going to mash it again. Really, really, really carefully. You need to make sure that's really well mashed. Again, if you want to use a pestle and mortar or whiz it with a handheld blender, get a grown-up to help you with that, and that'll work really, really well. You need to make sure you mash it for a lot longer than I just did, but I'm just demonstrating really for you. And here in the water bath is one I did earlier. So then you're going to make yourself a water bath. What you need to do is take your tub or your bowl and you need to put the kettle on, but turn it off before it's boiled. It needs to be about 60 degrees, so it's still quite hot, but not boiling. Pour it into your water bath and use a thermometer to check whether it's 60 degrees. If it's too hot, add a little bit of cold water. If it's too cold, put the kettle back on till it's boiling and add a little bit of boiling water. As I said, please get a grown-up to help you with that part. Add your mixture into your water bath. And you need to leave that now for 20 minutes. Don't do anything to it. After 20 minutes, the DNA should have been fully extracted from those cells, but it's part of the mixture. Now, we're not going to see it like that, and that is where our freezing cold methylated spirit or white rum is important. Before you start the experiment, put it in the freezer for about half an hour and it'll be perfect. Now, pour it very, very, very slowly and carefully down the side of your glass. Don't worry if you spill a little bit. The most important thing is you do it very, very, very slowly. And as you're pouring it, because the alcohol is freezing cold and it's not very dense, it floats on the top. And you will see a see-through layer on the top of your mixture forming. You'll probably want a thicker layer of rum. Add more rum or methylated spirit to the top until you've got a good thick layer. And then all you need to do is wait and watch. And you will start to see some DNA starting to move into that see-through layer. Now, what are you looking for? You're looking for something that looks a little bit like strings floating around in that clear liquid on the top. You can get a fork, and after a while, where it's all been condensing up in that layer, you can use your fork to fish a little bit out, and you will find it's a little bit like jelly. Now, this DNA is all clumped up in a big blob. And an interesting fact for you is that if you were to take all of the DNA out of the cell of a person, all the cells of a person's body, take out the DNA, stretch it out to one long string, and it would pass from Earth to the Sun and back again about 63 times. That's the DNA from just one person. And the incredible thing is, all that length of DNA, and you wouldn't be able to see it. It's so thin that even the most powerful microscope in the whole world would not be able to see it. So it would stretch to the Sun and back 63 times, but you wouldn't be able to see it because it's so thin. Incredible substance. It's everywhere, in every living thing existing in the world. And it's incredibly important because it makes you who you are. Have a go at extracting the DNA from a kiwi fruit, and please share with us what you've discovered. Have a great time. Bye.